Chapter 2: An Introduction to Genes and Genomes. Encoded within DNA are genes that provide instructions controlling the activities of all cells. Genes influence our behavior, determine our physical appearances such as skin, hair, and eye color, and affect our susceptibility to genetic disease conditions. After completing this chapter, you should be able to compare and contrast prokaryotic and eukaryotic cells. Discuss experiments that determined that DNA is the inherited genetic material of living organisms. Describe the structure of a nucleotide and explain how they form double helical DNA molecules. Describe the process of DNA replication and discuss the role of key proteins involved. Understand what genomes are and why biologists study them. Describe the process of transcription and understand how mRNA creates a functional mRNA molecule. Describe the process of translation, and the roles of mRNA, tRNA, and rRNA. Explain why gene expression regulation is important. Explain what the epigenome is and understand why epigenetics is important. Discuss the role of operons in regulating gene expression in bacteria. Name different types of mutations and give examples of the consequences of mutations. Central to the study of biotechnology is an understanding of the structure of DNA as the molecule of life, the inherited genetic material. Later, we will consider how extraordinary techniques in molecular biology enable biologists to clone and engineer DNA manipulations that are essential for many applications in biotechnology. Chapter 3. In this chapter, we review DNA structure and replication, discuss how genes code for proteins, provide an overview of genomics, and consider causes and consequences of mutations. Forecasting the future. Although much of what we will discuss in this chapter describes basic information about cell structure and genes that scientists have known about for decades, there are many exciting potential future developments especially related to genomics. An active area of research is the creation of artificial cells in which synthetic genomes are assembled in a lab and introduced into cells to create cells with novel properties based on the inserted genome. Similarly, another area of intense investigation is the creation of novel proteins by combining DNA sequences. These approaches have not yet advanced far enough to produce valuable applications. But there are signs that synthetic genome strategies for making cells with novel properties can work. Fundamental information about cells, gene expression, and genomes is playing a major role in the development of cell-based treatment strategies such as using stem cell to create tissues and organs, which we will discuss in Chapter 11. Advances in genetic testing and novel therapies designed to target human genes will continue to be an area of focus for biotechnology companies. 2.1a Review of cell structure. Cells are the structural and functional units of life. Organisms such as bacteria consist of a single cell, whereas humans have approximately 75 trillion, including over 200 different types that vary in appearance and function. Cells vary greatly in size and complexity, from tiny bacterial cells to human neurons that may stretch for more than 3 feet from the spinal cord to muscles in the toes. Virtually all cells share a common component, genetic information in the form of deoxyribonucleic acid, DNA. Genes control numerous activities in cells by directing the synthesis of proteins. Genes influence our behavior, determine our physical appearance, such as skin, hair, and eye color, and affect our susceptibility to genetic disease conditions. Before we begin our study of genes and genomes, we will review basic aspects of cell structure and function and briefly compare different types of cells. Prokaryotic cells. Cells are complex entities with specialized structures that determine cell function. Generally, every cell has a plasma, cell, membrane, a double-layered structure of primarily lipids and proteins that surrounds its outer surface, cytoplasm, the inner contents of the cell between the nucleus and the plasma membrane, and organelles, little organs, structures in the cell that perform specific functions. Throughout this book, we not only consider how plant and animal cells play important roles in biotechnology but also cover many biotechnology applications involving bacteria, yeasts, and other microorganisms. Bacteria are referred to as prokaryotic cells, or simply prokaryotes, from the Greek words meaning before nucleus, because they do not have a nucleus, an organelle that contains DNA in animal and plant cells. Prokaryotes include true bacteria, eubacteria, and cyanobacteria, a type of blue-green algae, Table 2.1, and members of the domain Archaea, ancient bacteria with some eukaryotic characteristics. Bacteria have a relatively simple structure, Figure 2.1. Their outer boundary is defined by the plasma membrane, which is surrounded by a rigid cell wall that protects the cell. Except for ribosomes, which are used for protein synthesis, bacteria have few organelles. The cytoplasm contains DNA, usually in the form of a single circular molecule, which is attached to the plasma membrane and located in an area called the nucleoid region, Figure 2.1. Some bacteria also have a tail-like structure called a flagellum, which is used for locomotion. Eukaryotic cells. Plant and animal cells are considered eukaryotic cells, from the Greek words meaning true nucleus, because they contain a membrane-enclosed nucleus and many organelles. Eukaryotes also include fungi and single-celled organisms called protists, which include most. Table 2.1 Prokaryotic and eukaryotic cells. Prokaryotic cells. Eukaryotic cells. Cell types Archaebacteria. True bacteria, eubacteria. Protists, fungi, plant, animal cells. Size: 100 nm 10 μm. 10 to 100 μm. Structure: no nucleus, DNA located in the cytoplasm. No organelles. DNA enclosed in a membrane-bound cytoplasm. Nucleus. Many organelles. Figure 2.1 Prokaryotic cell structure. Bacteria are prokaryotes. Shown here is a drawing of structures contained in a typical rod-shaped bacterium. Algae. Diagrams of plant and animal cells are shown in Figure 2.2. The plasma membrane is a fluid, highly dynamic, complex double-layered barrier composed of lipids, proteins, and carbohydrates. The membrane performs essential roles in cell adhesion, cell-to-cell communication, and cell shape, and it is essential for transporting molecules into and out of the cell. The membrane also serves an important role as a selectively permeable barrier, because it contains proteins involved in complex transport processes that control which molecules can enter and leave the cell. For example, hormones such as insulin are released from the cell in a process called secretion. Other molecules, such as glucose, can be taken into the cell and, within mitochondria, be converted into energy in the form of a molecule called adenosine triphosphate, ADP. Membranes also enclose or comprise many organelles. The cytoplasm of eukaryotes consists of cytosol, a nutrient-rich, gel-like fluid, and many organelles. The cytoplasm of prokaryotes also contains cytosol, but few organelles. Think of the organelle as the compartment in which chemical reactions and cellular processes occur. Organelles allow cells to carry out thousands of different complex reactions simultaneously. Each organelle is responsible for specific biochemical reactions. For instance, lysosomes break down foreign materials and old organelles. Organelles such as the endoplasmic reticulum and Golgi apparatus synthesize proteins, lipids, and carbohydrates, sugars. By compartmentalizing reactions, cells can carry out a multitude of reactions in a highly coordinated fashion simultaneously without interference. Be sure to familiarize yourself with the functions of organelles presented in Figure 2.2 and Table 2.2. Figure 2.2 Eukaryotic cell structure. Sketches of common structures present in plant, A, and animal cells, B. Table 2.2 Structure and Function of Eukaryotic Cells. Cell part. Structure. Functions. Plasma membrane. Membrane made of a double layer of lipids, primarily phospholipids and cholesterol, within which proteins are embedded. Proteins may extend entirely through the lipid bilayer or protrude on only one face. Externally facing proteins and some lipids have attached sugar groups. Serves as an external cell barrier, acts in the transport of substances into or out of the cell, maintains a resting potential that is essential for the functioning of excitable cells. Externally facing proteins act as receptors, for hormones, neurotransmitters, and so on, and in cell-to-cell recognition. Cytoplasm. Cellular region between the nuclear and plasma membranes, consists of fluid cytosol, containing dissolved solutes, inclusions, stored nutrients, secretory products, pigment granules, and organelles, the metabolic machinery of the cytoplasm. Cytoplasmic organelles. Mitochondria. Rod-like double membrane structures, the inner membrane is folded into projections called cristae. Site of adenosine triphosphate, ADP, synthesis, powerhouse of the cell. Ribosomes. Dense particles consisting of two subunits, each composed of ribosomal RNA and protein, free or attached to rough endoplasmic reticulum. The sites of protein synthesis. Rough endoplasmic reticulum. Membrane system enclosing a cavity, the cisterna, and coiling through the cytoplasm, externally studded with ribosomes. Sugar groups are attached to proteins within the cisternae. Proteins are bound in vesicles for transport to the Golgi apparatus and other sites. External face synthesizes phospholipids and cholesterol. Smooth endoplasmic reticulum. Membranous system of sacs and tubules, free of ribosomes. Site of lipid and steroid synthesis, lipid metabolism, and drug detoxification. Golgi apparatus. A stack of smooth membrane sacs and associated vesicles close to the nucleus. Packages, modifies, and segregates proteins for secretion from the cell, inclusion in lysosomes, and incorporation into the plasma membrane. Lysosomes. Membranous sacs containing hydrolases, digestive enzymes. Sites of intracellular digestion. Peroxisomes. Membranous sacs of oxidase enzymes. The enzymes detoxify a number of toxic substances. The most important enzyme, catalase, breaks down hydrogen peroxide. Microtubules. Cylindrical structures made of tubulin proteins. Support the cell and give it shape, involved in intracellular and cellular movements, form centrioles. Microfilaments. Fine filaments of the contractile protein actin. Involved in muscle contraction and other types of intracellular movement, help form the cell's cytoskeleton. Intermediate filaments. Protein fibers, composition varies. The stable cytoskeletal elements resist mechanical forces acting on the cell. Centrioles. Paired cylindrical bodies, each composed of 9 triplets of microtubules. Organize a microtubule network during mitosis to form the spindle and asters, form the bases of cilia and flagella. Cilia. Short, cell surface projections, each cilium is composed of 9 pairs of microtubules surrounding a central pair. Move in unison, creating a unidirectional current that propels substances across cell surfaces. Flagella. Like cilia but longer, the only example in humans is the sperm tail. Propels the cell. Nucleus. Largest organelle, surrounded by the nuclear envelope, contains fluid nucleoplasm, nucleoli, and chromatin. Control center of the cell, responsible for transmitting genetic information and providing the instructions for protein synthesis. Nuclear envelope. Double membrane structure pierced by the pores, outer membrane continuous with the cytoplasmic endoplasmic reticulum. Separates the nucleoplasm from the cytoplasm and regulates the passage of substances to and from the nucleus. Nucleoli. Dense spherical, non-membrane bound, bodies composed of ribosomal RNA and proteins. Site of ribosome subunit manufacture. Chromatin. Granular, thread-like material composed of DNA and histone proteins. DNA contains genes. Central vacuole, plant cells. Large membrane enclosed compartment. Used to store ions, waste products, pigments, protective compounds. Chloroplasts, plant cells. Membrane enclosed organelle containing stacked structures, grana, of chlorophyll containing membrane sacs called thylakoids surrounded by an inner fluid, stroma. Site of photosynthesis. The nucleus contains DNA. This organelle is a spherical structure enclosed by a double-layered membrane, the nuclear envelope, and is typically the largest structure in a eukaryotic cell. Nearly 6 feet of DNA is coiled into the nucleus of every human cell. If the DNA in all human cells were connected end to end, there would be enough to stretch to the sun and back about 500 times. Although the majority of DNA in a eukaryotic cell is contained within the nucleus, mitochondria and chloroplasts also contain small circular DNA molecules. 2.2 The molecule of life. Every high school or college biology course involves some discussion of DNA, and DNA is routinely manipulated by students in college biology laboratories and many high school classes. With the wealth of information available about many detailed aspects of DNA and genes, the study of biology in the 21st century might give you the impression that the structural details of DNA were always understood. However, the structure of DNA and its function as genetic material was not always well known. Many extraordinary researchers and incredible discoveries have contributed to our modern-day understanding of DNA structure and function. We begin this section with a brief overview, highlighting the evidence for DNA as the genetic material, then we discuss DNA's structure. Evidence that DNA is the inherited genetic material. In 1869, Swiss biologist Friedrich Miescher identified a cellular substance from the nucleus that he called nuclein. Miescher purified nuclein from white blood cells and found that it could not be broken down, degraded, by protein-digesting enzymes called proteases. This discovery suggested that nuclein was not made only of proteins. Subsequent studies determined that this material had acidic properties, which led nuclein to be renamed nucleic acids. DNA and ribonucleic acid, RNA, are the two major types of nucleic acids. While biochemists worked to identify the different components of nucleic acids, British microbiologist Frederick Griffith's experiments, 1928, with 2 strains of Streptococcus pneumoniae provided evidence that DNA is the inherited genetic material of cells. Griffith was studying 2 strains of the bacterium Streptococcus pneumoniae, a microbe that causes pneumonia. At the time of these studies, this strain was called Diplococcus pneumoniae. Griffith worked with a virulent, disease-causing, variety called the smooth. Figure 2.3 Griffith's transformation experiment. The S strain of S. pneumoniae kills mice. A. The R strain is harmless. B. Heat-killed S cells are harmless. C. Mice injected with heat-killed S cells mixed together with live R cells died. D. And live S cells could be detected in the blood of dead mice. E. This result is a demonstration of transformation. Living R cells took in DNA from dead S cells, transforming the R cells into S cells. Strain, S cells, along with a harmless strain called rough cells, R cells. S cells are surrounded by a capsule, smooth coat, of proteins and sugars, whereas R cells lack this coat. When Griffith injected mice with living S cells, the mice died, and Griffith found live S cells in the blood of the dead mice, figure 2.3. When live R cells were injected into mice, the mice lived and showed no living R cells in their blood. These experiments suggested that the protein coat was responsible for the death of the mice. To test this idea, Griffith then killed S cells by heating them, which destroys proteins in the coat. Not surprisingly, mice injected with heat-killed S cells lived, with no signs of live S cells in their blood. But when Griffith mixed heat-killed S cells with living R cells in a tube and injected this mixture into mice, the mice died and living S cells were found in the blood of the dead mice. Where did the live S cells come from? This experiment provided evidence that the genetic material from heat-killed S cells had transformed, changed, or converted R cells into S cells. Griffith's experiments demonstrated transformation, the uptake of DNA by bacteria. Heat treatment broke open S cells, which released their DNA into the tube. Living R cells took up S cell DNA, which transformed the properties of the R cells so that they became virulent, resembling S cells. As you will learn, transformation is a very powerful technique in molecular biology and is routinely used to introduce genes into bacteria for DNA cloning, protein production, and other important purposes. Although Griffith hypothesized that some genetic factor was responsible for the transformation results he saw, he didn't actually identify DNA as the transforming factor. However, his experiments were instrumental in leading others in search of this factor. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty purified DNA from large batches of S. pneumoniae grown in liquid culture. Their experiments provided definitive evidence that DNA is the genetic material and proved that DNA was the transforming factor in the Griffith experiments. In their now famous experiment, Avery, MacLeod, and McCarty ground up, homogenized, mixtures of bacterial cells from S. pneumoniae and treated these extracts with proteases, RNA-degrading enzymes, RNase, or DNA-degrading enzymes, DNase. They subsequently carried out transformation experiments using these treated extracts. Extracts from killed S cells were mixed with living R cells and injected into mice. This experiment demonstrated that DNase-treated extracts could not transform R cells to S cells because DNA in these mixtures was degraded by DNase. Extracts treated with protease or RNase still maintained their transforming ability because DNA in these mixtures remained intact. Although other studies with viruses were essential for determining the role of DNA, the work of Avery, MacLeod, and McCarty provided definitive evidence for DNA as the genetic material causing transformation. DNA structure. While evidence supporting DNA as hereditary material was building, a significant question still remained: what is the structure of DNA? Erwin Chargaff provided some insight to this question by isolating DNA from a variety of different species and revealing that the percentage of DNA bases called adenine was proportional to the percentage of bases called thymines, and that the percentage of cytosine bases in an organism's DNA were roughly proportional to the percentage of guanine. This valuable observation suggested that the bases adenine, thymine, cytosine, and guanine were somehow intricately related components of DNA structure, an important principle to remember because, as we explore next, these bases are essential components of DNA. The building block of DNA is the nucleotide, figure 2.4. Each nucleotide is composed of a 5-carbon, pentose sugar called deoxyribose, a phosphate molecule, and a nitrogenous base. The bases are interchangeable components of a nucleotide. Each nucleotide contains one base, either adenine, A, thymine, T, guanine, G, or cytosine, C, the so-called As, TS, GS, and CS of DNA. Figure 2.4 Nucleotide structure. All DNA nucleotides consist of a nitrogenous base, A, C, G, or T, a sugar, and a phosphate group. The sugar in DNA is deoxyribose. RNA molecules contain a pentose sugar called ribose. The pentose sugar in DNA is called deoxyribose because it lacks an oxygen at carbon number 2 of the sugar compared with the ribose sugar in RNA. A base is attached to carbon number 1 of the sugar, the phosphate group is attached to carbon number 5 of the sugar. Because of their structure, adenine and guanine belong to a group of bases called purines, whereas cytosine, thymine, and uracil belong to a group called pyrimidines. Nucleotides are the building blocks of DNA, but how are these structures arranged to form a DNA molecule? Many scientists have contributed to the answer to this question, but the definitive structure of DNA was finally revealed by James Watson and Francis Crick, working at the Cavendish Laboratories in Cambridge, England. Chemists Rosalind Franklin and Maurice Wilkins, of University College, London, used X-ray crystallography to provide Watson and Crick with invaluable data on the structure of DNA. By firing an X-ray beam onto crystals of DNA, Franklin and Wilkins revealed a model of DNA indicating that its structure could be helical. From these data, Chargaff's findings, and other studies, Watson and Crick assembled a wire model of DNA. Watson and Crick published The Molecular Structure of Nucleic Acids, a structure for deoxyribose nucleic acid in the prestigious journal Nature on April 25, 1953. The first paragraph of this paper reads, "We wish to suggest a structure for the salt of deoxyribose nucleic acid, DNA. This structure has novel features which are of considerable biological interest. Given the importance of DNA and what we have learned about DNA structure over the last 50 years, this description might be one of the greatest understatements ever made in a published scientific paper." The significance of this discovery was appropriately recognized in 1962, when Watson, Crick, and Wilkins received the Nobel Prize in Medicine. Watson and Crick determined that nucleotides form long strands of DNA and that each DNA molecule consists of two strands that join together and wrap around each other to form a double helix, figure 2.5. A strand of DNA is a string of nucleotides held together by phosphodiester bonds that connect the sugar of one nucleotide to the phosphate group of an adjacent nucleotide, figure 2.5. The sequence of bases in a strand can vary. For instance, a nucleotide containing AC can be connected to a nucleotide containing an A, T, G or another nucleotide containing AC. Each strand of nucleotides has a polarity to it. There is a 5' and a 3' to the strand, figure 2.5. Polarity refers to the carbons of the deoxyribose sugar. At the 5' of a strand, the phosphate at carbon 5 is not bonded to another nucleotide, but carbon 3 is involved in a phosphodiester bond. At the 3' the phosphate at carbon 5 is bonded to another nucleotide, but carbon 3 is not joined to another nucleotide. Although this aspect of nucleotide structure may seem trivial, the polarity of DNA is important for replication and for the routine manipulation of DNA in the laboratory. Watson and Crick determined that DNA molecules consist of two interconnecting strands that wrap around each other to form a right-handed double helix, perhaps the most famous molecular model in all of biology, figure 2.5. The two strands are joined together by hydrogen bonds between complementary base pairs in opposite strands, figure 2.5. Adenine base pairs with thymine, and guanine base pairs with cytosine. From this model, Chargaff's observations are easily understood. The proportions of A's and T's are equivalent in an organism's DNA, as are the proportions of G's and C's, because they pair with each other in a DNA molecule. The two strands of nucleotides in a double helix are antiparallel. The polarity of each strand is reversed relative to the other, figure 2.5. This orientation is necessary for the complementary base pairs to align and for hydrogen bonds to align with one another. The double helix resembles a twisted ladder of sorts. The rungs of this ladder consist of the complementary base pairs, and the sides of the ladder consist of sugar and phosphate molecules, creating the backbone of DNA. What is a gene? Genes are often described as units of inheritance, but what exactly is a gene? A gene is a sequence of nucleotides that provides cells with the instructions to synthesize a specific protein or a particular type of RNA. Not all genes are used to produce a protein. For example, genes for transfer RNA, tRNA, are used to make tRNA molecules, and while TRNAs are required for protein synthesis, they are not translated to produce a protein. Most genes are approximately 1,000 to 4,000 nucleotides, NT, long, although many smaller and larger genes have been identified. Largely by controlling the proteins produced by a cell, genes influence how cells, tissues, and organs appear, both through the microscope and with the naked eye. These inherited appearances are called traits. Through the DNA contained in your cells, you have inherited traits from your parents, such as eye color and skin color. Genes influence not only cell metabolism and behavioral and cognitive abilities such as intelligence but also our susceptibility to certain types of genetic diseases. Some traits are controlled by a single gene, but many others are determined by multiple genes, which produce many proteins that interact in complex ways. In section 2.4, we explore how genes direct protein synthesis in cells. Throughout this book, we consider examples of genes, their functions, and their many applications in different areas of biotechnology. 2.3 Chromosome structure, DNA replication, and genomes. Before we consider how genes function, it is important that you understand how and why DNA is organized into chromosomes and how DNA is replicated in cells. Figure 2.5 DNA is a double-stranded helix. A. Two strands of nucleotides are joined together by hydrogen bonds between complementary base pairs. Adenine bases, A, always base pair with thymine bases, T, and cytosine, C, base pairs with guanine, G. B. The two strands wrap around each other so that the overall structure of DNA is a double-stranded helix with a sugar phosphate backbone, in which the bases are aligned in the center of the helix. Chromosome structure. Suppose you were presented with a challenge. If you solved it, you would earn free tuition for the rest of your undergraduate courses. You are given a basket containing 46 packages of different colored yarn all unraveled and intertwined. Your challenge is to sort the yarn into 46 even balls. How would you solve this challenge? If you started to cut the tangled pile of yarn randomly, you probably would not succeed. Of course, if you painstakingly unraveled the yarn and wound each color of yarn into a ball, you would eventually sort it into 46 even balls. This analogy provides a highly simplified view of the challenge presented to a human cell when it has to divide and sort its DNA into even packages. The 3 billion base pairs, BP, of DNA in every human cell would stretch to around 6 feet if unraveled, an amazing amount of material packed into the tiny nucleus of each cell. This DNA must be separated evenly when a cell divides, otherwise the loss of DNA can have devastating consequences. Fortunately, such mistakes in DNA separation are rare, in part because cells effectively separate and package DNA into chromosomes. Inside the nucleus, DNA exists in a relatively unraveled state. This does not mean that the DNA is uncoiled from its double helical structure, rather, the DNA is somewhat loosely arranged and not fully compacted into tightly coiled chromosomes, although all of the DNA in a chromosome remains together within the nucleus. When a cell is not dividing, chromosomes in the nucleus exist as an intricate combination of DNA and DNA-binding proteins called histones to form strings called chromatin. During cell division, chromatin is coiled into tight fibers that eventually wrap around each other, so that chromosomes become highly coiled and tightly condensed packages of DNA and histones. Figure 2.6. The size and number of chromosomes vary from species to species. Most bacteria have a single circular chromosome, in the size range of several 100,000 base pairs, which contains a few thousand genes. Eukaryotes typically contain one or more sets of chromosomes, which have a linear shape, and often these chromosomes are several million base pairs in size. Most human cells have two sets, pairs, of 23 chromosomes each, for a total of 46 chromosomes. Through the process of fertilization, you inherited 23 chromosomes from your mother, maternal chromosomes, and 23 chromosomes from your father, paternal chromosomes. These chromosome pairs are called homologous pairs, or homologs. Chromosomes 1 through 22 are known as the autosomes. The 23rd pair are called the sex chromosomes, consisting of X and Y chromosomes. Human egg and sperm cells, called the sex cells or gametes, contain a single set of 23 chromosomes, called the haploid number, N, of chromosomes. All other cells of the body, such as skin cells, muscle cells, and liver cells, are known as somatic cells. Somatic cells from many organisms have two sets of chromosomes, called the diploid number, 2N, of chromosomes. Human somatic cells contain 46 chromosomes. Somatic cells of a normal human male have 22 pairs of autosomes and an X and Y chromosome. Cells of a normal female have 22 pairs of autosomes and 2 X chromosomes. Sex chromosomes were so named because they contain genes that influence sex traits and the development of reproductive organs, whereas the autosomes were originally thought primarily to contain genes that affect body features unrelated to sex, such as skin color and eye color. Although there are genes involved in sex organ determination that are present on the Y. Figure 2.6 Chromosome organization. In a non-dividing cell, chromosomal DNA exists in an unraveled state called chromatin. Histone proteins serve as particles around which DNA becomes tightly wound to give a beads on a string appearance when viewed with an electron microscope. When cells divide, chromatin is further compacted into tight fibers and supercoiled looped structures. Ultimately, these supercoiled loops are tightly packed together with the assistance of other proteins to create an entire chromosome, a highly compact assembly of DNA. Each chromosome consists of two sister chromatids attached by a centromere. Chromosome arms are the portions of the chromatid on one side of the centromere, labeled as the P and Q arms. The ends of a chromosome are called telomeres. Chromosome. There are other genes involved in sex determination that are present on autosomes, and the majority of genes on the X chromosome are not required for development of the reproductive organs. Several characteristics are common to most eukaryotic chromosomes. Each chromosome consists of two thin, rod-like structures of DNA called sister chromatids, figure 2.6. The sister chromatids are exact replicas of each other, copied during DNA synthesis. During cell division, sister chromatids are separated so that newly forming cells receive the same amount of DNA as the original cell from which they arose. Each eukaryotic chromosome has a single centromere, a constricted region of the chromosome consisting of intertwined DNA and proteins that join the two sister chromatids to each other. This region of a chromosome also contains proteins that attach chromosomes to organelles called microtubules, which play an essential role in moving chromosomes and separating sister chromatids during cell division. The centromere delineates each sister chromatid into two arms: the short arm, called the P arm, and the long arm, or Q arm. Each arm of a chromosome ends with a segment called a telomere, figure 2.6. Telomeres are highly conserved repetitive sequences of nucleotides that are important for attaching chromosomes to the nuclear envelope. Telomeres are a subject of intense research. Changes in telomere length are believed to play a role in the aging process and in the development of certain types of cancers. Karyotype analysis for studying chromosomes. One most common way to study chromosome number and basic aspects of chromosome structure is to prepare a karyotype. In karyotype analysis, cells are spread on a microscope slide and then treated with chemicals to release and stain the chromosomes. For example, G-banding, in which chromosomes are treated with a DNA binding dye called Giemsa stain, creates a series of alternating light and dark bands in stained chromosomes. Each stained chromosome shows a unique and reproducible banding pattern that can be used to identify different chromosomes. Chromosomes can be aligned and paired based on their staining pattern and size, figure 2.7. In humans, chromosome 1 is the largest chromosome. Figure 2.7 Karyotype analysis. In a karyotype, dividing cells are spread out onto a glass microscope slide to release their chromosomes. Chromosomes are stained and aligned based on their overall size, the position of the centromere, and their staining pattern to create a karyotype. And chromosome 21 is the smallest. Karyotypes are very valuable for studying and comparing chromosome structure. A more modern method for karyotype analysis, called spectral karyotyping, incorporates specific probes and techniques to colorize chromosomes. It provides a more detailed analysis of chromosome structure than traditional karyotypes, such as the one shown in figure 2.7. Karyotype analysis can be used to identify human genetic disease conditions associated with abnormalities in chromosomal structure and number. DNA replication. When a cell divides, it is essential that the newly created cells contain equal copies of replicated DNA. Somatic cells divide by a process called mitosis, wherein one cell divides to produce daughter cells, each of which contains an identical copy of the DNA of the original, parent, cell. For instance, a human skin cell divides to produce two daughter cells, each containing 23 pairs of chromosomes. Gametes are formed by a process called meiosis, wherein a parent cell divides to create up to 4 daughter cells, which can be either sperm or egg cells. During meiosis, the chromosome number in daughter cells is cut in half to the haploid number. Sperm and egg cells each contain a single set of 23 chromosomes. Through sexual reproduction, a fertilized egg, called the zygote, is formed. The zygote, which divides by mitosis to form an embryo and eventually a complete human, contains 46 chromosomes, 23 paternal chromosomes and 23 maternal chromosomes. Prior to cell division by either mitosis or meiosis, DNA must be replicated in the cell. Replication occurs by a process called semi-conservative replication. Figure 2.8 shows an overview of this process. Before replication begins, the two complementary strands of the double helix must be pulled apart into single strands. Once separated, these strands serve as templates for copying two new strands of DNA. At the end of this process, two new double helices are formed. Each helix contains one original DNA, parental, strand and one newly synthesized strand, thus the term semi-conservative. DNA replication occurs in a series of stages involving a number of different proteins. Because prokaryotes. Figure 2.8 An overview of DNA replication. Nucleotide strands in a DNA molecule must first be separated, A. Each strand serves as a template for the synthesis of new strands, producing two DNA molecules, each containing one original strand and one newly synthesized strand, B. Figure 2.9 Semi-conservative replication of DNA. Contain circular chromosomes. DNA replication in prokaryotes is slightly different from that in eukaryotes. Here we consider the main players and key concepts in DNA replication overall. But keep in mind that there are subtle differences distinguishing the replication process in prokaryotes from that in eukaryotes. Replication is initiated by DNA helicase, an enzyme that separates the two strands of nucleotides, literally unzipping the DNA by breaking hydrogen bonds between complementary base pairs, figure 2.9. The separated strands form a replication fork. As helicase unwinds the DNA and single-strand binding proteins attach to each strand and prevent them from base pairing and reforming a double helix. This step is important because the DNA strands must be held apart during DNA replication. Strand separation occurs at sites called origins of replication. Bacterial chromosomes have a single origin. Because of their large size, eukaryotic chromosomes have multiple origins. Starting DNA replication at multiple origins allows eukaryotic chromosomes to be copied rapidly. The next step in DNA replication involves the addition of short segments of RNA approximately 10 to 15 nucleotides long. These sequences, called RNA primers, are synthesized by an enzyme called primase. In eukaryotes, a form of DNA polymerase called alpha-alpha acts as the primase. Primers start the process of DNA replication because they serve as binding sites for DNA polymerases, the key enzymes that synthesize new strands of DNA. Several different forms of DNA polymerase are involved in copying DNA. In bacteria, the enzyme DNA polymerase III, called DNA polymerase delta-delta in eukaryotes, binds to each single strand, moving along the strand and using it as a template to copy a new strand of DNA. During this process, DNA polymerase uses nucleotides present in the cell to synthesize complementary strands of DNA. DNA polymerase always works in one direction, synthesizing new strands in a 5:3 orientation and adding nucleotides to the 3 end of a newly synthesized strand, figure 2.9, by forming phosphodiester bonds between the phosphate of one nucleotide and the sugar in the previous nucleotide. Because DNA polymerase proceeds only in a 5:3 direction, replication along one strand, the leading strand, occurs in a continuous fashion, figure 2.9. Synthesis on the opposite strand, the lagging strand, occurs in a discontinuous fashion because DNA polymerase must wait for the replication fork to open. On the lagging strand, short pieces of DNA, called Okazaki fragments, named after Reiji and Tunko Okazaki, the scientists who discovered these fragments, are synthesized as the DNA polymerase works its way out of the replication fork. Covalent bonds between Okazaki fragments in the lagging strand are formed by DNA ligase to ensure that there are no gaps in the phosphodiester backbone. Finally, the RNA primers are removed and these gaps are filled by DNA polymerase. Remember the functions of enzymes involved in DNA synthesis. As we will discuss, DNA polymerase and DNA ligase are routinely used in the lab in working with cloned DNA, Chapter 3. What is a genome? DNA contains the instructions for life genes. All of the DNA in an organism's cells is called the genome. Contained in the human genome are approximately 20,000 genes scattered among 3 billion base pairs of DNA. The study of genomes, a discipline called genomics, is currently one of the most active and rapidly advancing areas of biological science. Throughout this book, we discuss aspects of the Human Genome Project, a worldwide effort to identify all human genes on each chromosome. The Human Genome Project was an enormous undertaking in genomics that has provided exciting insight into human genes, their locations, and their functions. 2.4 RNA and protein synthesis. Genes govern the activities and functions within a cell by directing the synthesis of proteins. Some of the myriad functions of these essential molecules are as follows. Proteins are necessary for cell structure, for example, as important components of membranes, organelles, and the cytoplasm. Proteins carry out essential reactions in the cell as enzymes. Proteins perform critical roles as hormones and other signaling molecules that cells use to communicate with one another. Receptor proteins bind to other molecules, such as hormones, and transport proteins, enabling molecules to enter and leave cells. Proteins as antibodies recognize and destroy foreign materials in the body. Quite simply, cells cannot function without proteins. How does DNA make proteins? Actually, DNA does not make proteins directly. To synthesize proteins, genes are first copied into molecules called messenger RNA, mRNA, figure 2.10. RNA synthesis is called transcription, because genes are literally transcribed, copied, from a DNA code into an RNA code. In turn, mRNA molecules, which are exact copies of genes, contain information that is deciphered into instructions for making a protein through a process known as translation. RNA molecules are single-stranded, not double-stranded like DNA, but the chemical composition of RNA is very similar to that of DNA. The bases of RNA are also very similar to those of DNA. One key difference is that RNA contains a base called uracil, U, instead of thymine, T, see figure 2.4. The other primary difference is that RNA contains a pentose sugar called ribose, which has a slightly different structure than the deoxyribose sugar contained in DNA. Figure 2.10 The flow of genetic information in cells. DNA is copied into RNA during the process of transcription. RNA directs the synthesis of proteins during translation. Through proteins, genes control the metabolic and physical properties or traits of an organism. An easy way to remember the difference between transcription and translation is to remember that translation involves a change in code from RNA to protein, much like translating one language to another. Through the production of mRNA and the synthesis of proteins, DNA controls the properties of a cell and its traits, figure 2.11. This process of transcription and translation directs the flow of genetic information in cells, controlling a cell's activities and properties. Here we study basic principles of transcription and translation and aspects of how gene expression can be controlled by cells. Copying the code, transcription. How is DNA used as a template to make RNA? RNA polymerase is a key enzyme for transcription. Inside the nucleus, RNA polymerase unwinds the DNA helix and then copies one strand of DNA into RNA. Unlike DNA replication, in which the entire DNA molecule is copied, transcription occurs only in segments of a chromosome that contain genes. How does RNA polymerase know where to begin transcription? Tools of the trait enzymes involved in DNA replication have valuable and essential roles in molecular biology research. This chapter introduces important principles of DNA structure, replication, transcription, and translation and provides an essential background on genes and genomes. These principles are important not only for understanding what genes are and how they function but also many of the components involved in processes such as DNA replication, which have become essential tools for research in molecular biology. In the next chapter, you will learn how scientists can identify, clone, and analyze genes, a fascinating aspect of molecular biology research and biotechnology. The technology for cloning and studying genes became possible only as scientists learned more about the enzymes involved in processes such as DNA replication. For instance, DNA polymerase, the key enzyme that synthesizes DNA during semi-conservative replication, is widely used in molecular biology labs to copy DNA. Similarly, RNA polymerase is also widely used in molecular research. In addition, many DNA cloning procedures rely on DNA ligase to join together DNA fragments from different sources, a process called recombinant DNA technology. Because most of these enzymes are relatively inexpensive and readily available from supply companies, their use has become commonplace in most molecular biology labs. Without our understanding of the enzymes that act on DNA and RNA and their functions, many modern applications of biotechnology and a majority of techniques routinely used in molecular biology research would be impossible. Figure 2.11 Transcription. During transcription, RNA polymerase binds to DNA at a promoter region adjacent to a gene sequence and unwinds the DNA. RNA polymerase moves along the DNA template, copying one strand into a molecule of RNA. When RNA polymerase reaches a termination sequence, it releases from the DNA and transcription ends. Adjacent to most genes is a promoter, specific sequences of nucleotides that allow RNA polymerase to bind at specific locations next to genes. Proteins called transcription factors help RNA polymerase find the promoter and bind to DNA. Sequences called enhancers can also play important roles in transcription. After RNA polymerase binds to a promoter, it unwinds a region of the DNA to separate the two strands. Only one of the strands, called the template strand, the opposite strand is called the coding strand, is copied by RNA polymerase. The presence of a promoter determines which strand of DNA will be transcribed. Once properly oriented, RNA polymerase proceeds along the DNA template strand to copy a complementary strand of RNA by forming phosphodiester bonds between ribonucleotides in a 5:3 direction, figure 2.11. When RNA polymerase reaches the end of a gene, it encounters a termination sequence. These sequences bind either specific proteins or base pairs to create loops at the end of the RNA. As a result, the RNA polymerase and newly formed RNA are released from the DNA molecule. Unlike DNA replication, in which the DNA is copied only once each time a cell divides, multiple copies of mRNA are transcribed from each gene during transcription. Sometimes a cell transcribes thousands of copies of mRNA from a gene. Later in this section, you will see that cells with a high requirement for a particular protein generally produce large amounts of mRNA to encode that protein. Transcription produces different types of RNA. We have already seen that mRNA is produced when many genes are copied into RNA. Two other types of RNA, transfer RNA, tRNA, and ribosomal RNA, rRNA, are also produced by transcription. Different RNA polymerases produce each type of RNA. As we will soon learn, only mRNA carries information that directly codes for the synthesis of a protein, but tRNA and rRNA are also essential for protein synthesis. It is only relatively recently that scientists discovered a new class of non-protein coding RNA molecules called microRNAs, mRNAs. microRNAs are part of a rapidly growing family of small RNA molecules about 20 to 25 nucleotides in size that play novel roles in regulating gene expression. Later we'll briefly discuss the role of mRNAs. mRNA processing. In eukaryotic cells, the initial mRNA copied from a gene is called a primary transcript, pre-mRNA. This mRNA is immature and not fully functional. Primary transcripts undergo a series of modifications, collectively called mRNA processing, before they are ready for protein synthesis. One modification involves RNA splicing, figure 2.12. When the details of transcription were first worked out, scientists were surprised to learn that genes are interrupted by stretches of DNA that do not contain protein coding information, called introns. Introns are interspersed between exons, or protein coding sequences of a gene. Introns and exons are copied during transcription of mRNA. Before mRNA can be used to make a protein, the exons must be spliced together. As a simple analogy, think of introns as randomly inserted letters in a sentence that must be removed before the sentence can make sense. In the splicing process, introns are cut out of the primary transcript and adjacent exons are spliced to form a fully functional mRNA molecule with no introns. Splicing provides flexibility in the types of proteins that can ultimately be produced from a single gene. When genes were first discovered, scientists thought that a single gene could produce only one protein. But as the process of splicing was revealed, it became clear that when a gene contains several exons, splicing doesn't always occur in the same way. As a result, multiple proteins can be produced from a single gene. In a complex process called alternative splicing, splicing can sometimes join together certain exons and cut out other exons, essentially treating them as introns, figure 2.12b. This process creates multiple mRNAs of different sizes from the same gene. Each mRNA can then be used to produce different proteins with different, sometimes unique functions. Alternative splicing allows several different protein products to be produced from the same gene sequence. For instance, certain genes used to produce antibodies are alternatively spliced to produce some antibodies that attach to the surface of cells as well as other antibodies with different structures causing them to be secreted into the bloodstream. Similar splicing occurs with neurotransmitter genes, among many others. In fact, scientists originally believed that the human genome contained approximately 100,000 genes based on a predicted number of proteins made by human cells. As you will learn, genome scientists were very surprised to find that the human genome contains only about 20,000 genes. Chapter 3. Much of the reason for this discrepancy is that many human genes can be spliced in different ways, and estimates suggest that over 90% of human genes may use alternative splicing to create multiple different RNAs and proteins. Another type of processing occurs at the 5' end of mRNA, where a guanine base containing a methyl group is added. Figure 2.12. Known as a 5-cap, this structure plays a role in ribosome recognition of the 5' end of the mRNA molecule during translation. Figure 2.12. A eukaryotic gene and mRNA processing. A. Transcription of a eukaryotic gene produces a primary transcript or pre-mRNA, which undergoes processing through RNA splicing, the addition of a 5-cap, and polyadenylation. After processing, the final, mature mRNA is ready for export to the cytoplasm, where it will be translated into a protein. B. Alternative splicing can produce different RNAs and protein products from the same gene. Notice that the larger mRNA on the left contains 3 exons spliced together, but that the shorter mRNA on the right contains only 2 exons spliced together. Last, in a process called polyadenylation, a string of adenine nucleotides around 100 to 300 nucleotides in length is added to the 3' end of the mRNA, creating a tail, figure 2.12. This tail protects mRNA from RNA-degrading enzymes in the cytoplasm, increasing its stability and availability for translation. Following processing, a mature, processed mRNA molecule leaves the nucleus and enters the cytoplasm, where it is now ready for translation. Figure 2.12. Translating the code, protein synthesis. The ultimate function of a protein-coding gene is to produce a protein. Here we look at a brief overview of translation, using information in mRNA to synthesize a protein from amino acids. Translation occurs in the cytoplasm of cells as a multi-step process that involves several different types of RNA molecules. It will be much easier for you to understand the details of translation if you are familiar with important functions of each type of RNA. These are the major components of translation. Messenger RNA, mRNA, a copy of a gene, acts as a messenger by carrying the genetic code, encoded by DNA, from the nucleus to the cytoplasm, where this information can be read to produce a protein. Messenger RNAs vary in length from approximately 1,000 to several thousand nucleotides. Ribosomal RNA, rRNA, short single-stranded molecules around 1,500 to 4,700 nucleotides long. Ribosomal RNAs are important components of ribosomes, organelles that are essential for protein synthesis. Ribosomes recognize and bind to mRNA and read the mRNA during translation. Transfer RNA, tRNA, molecules that transport amino acids to the ribosome during protein synthesis. Transfer RNA molecules are approximately 75 to 90 nucleotides in length. We have already discussed the details of mRNA structure, but before we explore the details of translation, you need to be familiar with the genetic code of mRNA and the specific structures of ribosomes and tRNA. The genetic code. What is the genetic code contained within mRNA? As you will learn shortly, ribosomes read the code and then produce proteins, which are formed by joining the building blocks called amino acids. See Appendix 2. A chain of amino acids linked together by covalent bonds is a polypeptide. Some proteins consist of a single polypeptide chain, others contain several polypeptide chains that must wrap and fold around each other to form complicated three-dimensional structures. See Figure 4.3. Proteins can contain combinations of up to 20 different amino acids, yet there are only 4 bases in mRNA molecules. So how does this code work? What information is the ribosome decoding to tell a cell what amino acids belong in a protein? If there are only 4 nucleotides in mRNA, how can mRNA provide information coding for 20 different amino acids? The answers to these questions lie in the genetic code, a universal language of genetics used by virtually all living organisms. The code works in 3 nucleotide units called codons, which are contained within mRNA molecules. Each codon codes for a single amino acid, Table 2.3. For instance, notice that the codon UAC codes for the amino acid tyrosine, and the codon UGC codes for the amino acid cysteine. Although each codon codes for one amino acid, there is flexibility in the genetic code. There are 64 different potential codons corresponding to all possible combinations of the 4 possible bases assembled into 3 nucleotide codons, 43. But because there are only 20 amino acids, most amino acids may be coded for by more than one codon. For example, notice in Table 2.3 that the amino acid lysine may be coded for by AAA and AAG. Having a redundancy of codons increases the efficiency of translation. Some codons are present in RNAs with greater frequency than others, just as some words in the English language are preferred over others with identical meanings. Table 2.3 The genetic code. Also contained in the genetic code are codons that tell ribosomes where to begin or end translation. The start codon, OG, codes for the amino acid methionine and signals the starting point for mRNA translation. As a result, the first amino acid in many proteins is methionine, although this amino acid is removed shortly after translation in some proteins. Stop codons terminate translation. UGA is a commonly used stop codon, but UAA and UAG are other stop codons, Table 2.3. Stop codons do not code for amino acids, they simply signal the end of translation. The genetic code is universal. It is used by cells in humans, bacteria, plants, earthworms, fruit flies, and all other species. There are some subtle differences to the code in certain species, but at the basic level it operates the same way throughout biology. Because of this, biologists can use techniques called recombinant DNA technology to clone a human gene such as the insulin gene and insert it into bacteria so that bacterial cells transcribe and translate insulin, a protein they normally do not produce. Another helpful aspect of the universal genetic code is that it enables scientists to clone a gene in one species, such as a mouse, and then use sequence information from the mouse gene to identify a similar gene in humans. Because different species share a common genetic code, this approach is a very common strategy for identifying human genes, including many involved in disease processes. Ribosomes and tRNA molecules. Ribosomes are complex structures consisting of aggregates of rRNA and proteins that form structures called subunits. Each ribosome contains 2 subunits, the large and small subunits. These subunits associate to form 2 grooves, called the A, amino acyl, site and the P, peptidyl, site, into which tRNA molecules can bind, and an E site through which tRNA molecules leave the ribosome, figure 2.13. Transfer RNAs are small molecules less than 100 nucleotides long. Transfer RNAs fold in intricate ways. Figure 2.13 Stages of protein synthesis. A. Each ribosome contains a large and a small subunit. Shown here is a ribosome attached to mRNA. Abbreviated steps of translation are shown in steps 1 to 4. B. Diagrammatic example of the tRNA symbol used in this book. At one end of each tRNA is an amino acid binding site and at the opposite end is a 3-nucleotide anticodon sequence. And certain nucleotides in a tRNA base pair with each other. As a result, a tRNA assumes a structure called a cloverleaf, because as regions of the molecule base pair, other unpaired segments create loops. At one end of each tRNA is an amino acid attachment site, figure 2.13b. Enzymes in the cytoplasm called aminoacyl tRNA synthetases attach a single amino acid to each tRNA molecule, creating what is known as an aminoacyl transfer RNA, tRNA, or charged tRNA. Amino acyl tRNA molecules carry their amino acids to the ribosome and bind within grooves of the ribosomes at the A site. At the opposite end of each tRNA molecule is a 3-nucleotide sequence called an anticodon. Different amino acids have different anticodon sequences. Anticodons are designed to complementary base pair with codons in mRNA. Now that you know the players of translation mRNA, ribosomes, and tRNA, we will examine how these components come together to produce a protein. Stages of translation. There are some fundamental differences between translation in prokaryotes and eukaryotes. Here we provide an overview of basic aspects of the three major stages of translation in eukaryotes: initiation, elongation, and termination. The beginning of translation is called initiation. During initiation, the small ribosomal subunit binds to the 5' end of the mRNA molecule by recognizing the 5' cap of the mRNA. Other proteins, called initiation factors, are also involved in guiding the small subunit to the mRNA. The small subunit moves along the mRNA until it encounters the start codon OG. Pausing at the start codon, the small subunit waits for the correct tRNA, called the initiator tRNA, to come along. Figure 2.13. This tRNA has the amino acid methionine, MET, attached to it. Remember that most proteins begin with this amino acid, and contains the anticodon UAC. The UAC anticodon binds to the start codon by complementary base pairing, figure 2.13, then the large ribosomal subunit binds to this complex containing the small subunit, initiation factors, mRNA, and initiator tRNA. Once these components are in place, the ribosome can start translating a protein. The next step of translation is called elongation, because during this phase additional trans enter the ribosome, one at a time, and a growing polypeptide chain is elongated. The ribosome, paused at the second codon, waits for the second tRNA to enter the A site. In figure 2.13, notice that the second codon is UUC, which codes for the amino acid phenylalanine, PHE. The PHE trna enters the A site of the ribosome and the anticodon, AAG, base pairs with the codon. After two trnas are attached to the ribosome, an enzyme in the ribosome called peptidyltransferase catalyzes the formation of a peptide bond between the amino acids, attached to their trnas. Peptide bonds join together amino acids to form a polypeptide chain. After the amino acids are attached to each other, the initiator tRNA, without methionine attached, pauses briefly in the E site and then is released from the ribosome. Released empty trnas are recycled by the cell. A new amino acid is attached to the tRNA to create an amino acyl tRNA, which can be used again for translation. The newly forming polypeptide remains attached to the tRNA in the A site. During a phase called translocation, the ribosome shifts so the tRNA and growing protein move into the P site of the ribosome. The tRNA with a growing polypeptide chain attached is called a peptidyl tRNA. The A site of the ribosome is now aligned with the third codon in sequence, UG, which codes for tryptophan, and the ribosome waits for the proper amino acyl tRNA to enter the A site. The cycle continues as described to attach the next amino acid, tryptophan, to the growing protein and repeats itself as the ribosome moves along the mRNA. Elongation cycles continue to form a new protein until the ribosome encounters a stop codon, for instance, UGA. This signals the third stage of translation called termination. Remember that stop codons do not code for an amino acid. Proteins called releasing factors interact with the stop codon to terminate translation. The ribosomal subunits come apart and release from the mRNA, and the newly synthesized protein is released into the cell. Ribosomes do recycle and subsequently can bind to any other mRNA molecule, not just the mRNA for one particular gene, and start the process of translation again. Basics of gene expression control. The term gene expression refers to the production of mRNA, and sometimes protein, by a cell. Cells are exquisitely effective at controlling gene expression and translation to accommodate their needs. All genes are not transcribed and translated at the same rate in all cells. Gene expression is a dynamic process. Individual genes can be actively transcribed for a particular period of time, followed by periods of relatively little transcription depending on the needs of a cell. In addition, all cells of an organism contain the same genome, so how and why are skin cells different from brain cells or liver cells? Different cell types have different properties and carry out different functions because cells can regulate or control the genes they express. At any given time in a cell, only certain genes are turned on or expressed to produce proteins, while many other genes are silenced or repressed. These genes may be expressed by cells only at certain times, in response to specific cues from inside or outside of the cell, to make proteins as needed. These cues can be environmental. You decide access to biotechnology products for everyone? Later, you will learn how genes can be identified, cloned, and studied in great detail. Chapter 3. One benefit of gene cloning has been the identification of genes involved in human diseases. As a result, it is possible to make many gene products in the laboratory and use them for medical purposes. For instance, when the gene for insulin was cloned in bacteria, it became possible to produce large amounts of insulin for treating people with diabetes. Similarly, cloning of the gene for human growth hormone, HGH, which stimulates growth of bones and muscles during childhood, provided a readily available source of this hormone. Legally available by prescription only, HGH is used widely and effectively to treat children with certain forms of short stature or dwarfism. Illegal use of HGH by professional athletes has received much attention in recent years. Dwarfism is generally defined as a condition that results in an adult height of 4 feet 10 inches or shorter. The availability of HGH and other products of biotechnology raises an ethical question. Should HGH be available to everyone who wants taller children or only those children with dwarfism? Suppose parents wanted their average-size son to be taller so that he would have a better chance of making his high school varsity basketball team. Should these parents be able to give their son HGH simply to enhance his height? You decide. Signals such as temperature changes, nutrients in the external environment, hormones, or other complex chemical signals exchanged by cells. How can genes be turned on and off in response to different signals? Biologists call this process gene regulation. Prokaryotic cells and eukaryotic cells regulate gene expression in a variety of complex ways. Figure 2.14. One common mechanism used by both types of cells is called transcriptional regulation, controlling the amount of mRNA transcribed from a particular gene as a way to turn genes on or off. Here we provide an introduction to transcriptional regulation and consider basic examples of this process in eukaryotes and prokaryotes. Transcriptional regulation of gene expression. Because the amount of protein translated by a cell is often directly related to the amount of mRNA in the cell, cells can regulate the amount of mRNA produced for any given gene to control indirectly the amount of protein a cell produces. How do cells know which genes to turn on and which to shut off? To understand transcriptional regulation, we must look at the role of promoter sequences more closely. Figure 2.14 Levels of gene expression regulation. Prokaryotic and eukaryotic cells can regulate gene expression in a variety of complex ways. This figure summarizes the primary ways in which gene expression can be regulated in eukaryotic cells. Notice that gene expression regulation can occur at many different levels, highlighted in blue boxes here, beginning with how chromatin is folded or chemically modified to controlling degradation or turnover of a protein once it is made. Transcriptional regulation is a commonly used control mechanism in both prokaryotic and eukaryotic cells. Promoters are found upstream of gene sequences, meaning that they are found at the 5' end of a gene. Genes in prokaryotes and eukaryotes do not all use the same promoter sequences. In eukaryotes, common promoter sequences found upstream of many genes include ATATA box, TATA, located about 30 nucleotides, 30, upstream of the start site of a gene, and a CAAT box, GOT, located about 80 nucleotides, 80, upstream of a gene, figure 2.15. Earlier in this chapter we discussed how RNA polymerase initiates transcription by binding to promoter sequences adjacent to genes. For most eukaryotic genes, RNA polymerase cannot recognize and bind to a promoter unless transcription factors are present at the promoter. Transcription factors are DNA-binding proteins that can bind promoters and interact with RNA polymerase to stimulate transcription of a gene, figure 2.15. In eukaryotes and prokaryotes, common transcription factors interact with promoters for many genes. However, both types of cells also use specific transcription factors that interact only with certain promoters. Transcription of some genes also depends on the binding of specific transcription factors to regulatory sequences adjacent to the promoter. In addition, many genes that are tightly regulated by cells also contain regulatory sequences called enhancers. Enhancer sequences are usually located around 50 or more base pairs upstream of the promoter, but they can also be located downstream of a gene. Enhancer sequences bind regulatory proteins, generally referred to as activators. Activator molecules interact with transcription factors in RNA polymerase, forming a complex that stimulates, activates, transcription of a gene. Cells use a wide variety of different activator molecules. Each activator binds to a particular enhancer sequence. Some activator molecules can be hormones. For instance, the male sex steroid testosterone can be an activator to stimulate gene expression. You may know that testosterone stimulates cellular activities such as muscle and hair growth in developing boys, but how does testosterone work? This hormone binds to a receptor protein inside cells. The testosterone receptor protein complex acts as an activator to bind to a specific enhancer element in DNA called an androgen response element, 5-TGTTCT3. These elements are usually found close to a promoter. In turn, testosterone and its receptors stimulate gene expression. The female sex steroid estrogen works in a similar manner. Figure 2.15 promoters, transcription factors, and enhancers. But testosterone and other activators don't stimulate expression of all genes in all cells. Activators act only on those genes that contain enhancer sequences to which they can bind. Testosterone stimulates expression of genes involved in muscle and hair growth because these genes contain androgen response elements. Transcription of other genes without androgen response elements are not directly affected by the hormone. Incidentally, steroid abuse by bodybuilders and athletes looking to increase muscle mass and tone can cause serious long-term health effects, in part because steroids abnormally stimulate gene expression for prolonged periods of time. Through activators and enhancers, cells can use transcriptional control to regulate gene expression and control cellular activities. Some genes even contain repressor sequences that decrease transcription. Because different cells produce different transcription factors and activator molecules, genes can be turned on in some tissues and not others. Skin cells turn on different genes than muscle cells do, so each cell type produces different proteins, giving each cell type different functions. Consequently, tissue and cell-specific gene expression is one way for cells to control the proteins they express even though all body cells contain the same genome. These important control mechanisms are part of why different cells have different functions. In addition, identifying the promoters, enhancer sequences, and transcription factors that bind to regulatory regions of a gene is important for making biotechnology products. For instance, the identification of transcription factors that stimulate the expression of proteins needed for bone growth and development is helping scientists develop new drugs that can be used to stimulate bone growth in people suffering from forms of arthritis when their cells no longer produce the factors that stimulate bone growth. Bacteria use operons to regulate gene expression. Bacteria are very important organisms for many applications of biotechnology, such as the production of human proteins. Many initial studies on gene regulation were carried out in bacteria. Bacteria and other microorganisms can and must rapidly control gene expression in response to environmental stimuli such as growth nutrients and changes in temperature and light intensity. One interesting aspect of gene expression and regulation in bacteria is that many bacterial genes are organized in arrangements called operons. Operons are essentially clusters of several related genes located together and controlled by a single promoter. The genes of an operon can be regulated in response to changes within the cell. Operons can be stimulated, induced, or inhibited, repressed, depending on the needs of a cell. Many genes controlling nutrient metabolism by bacteria are organized as operons. Bacteria can use operons to tightly regulate gene expression in response to their nutrient requirements. Here we present a well-studied classic example of gene regulation in bacteria by describing the lac operon, Figure 2.16. Figure 2.16 The lac operon. By controlling the lac operon, bacterial cells can regulate gene expression in response to availability of the sugar lactose. In the absence of lactose, the lac repressor binds to the operator, blocking its transcription. In the presence of lactose, lactose binds and inactivates the repressor, allowing transcription of the operon to occur. The lac operon consists of the following three genes. Lax, encoding the enzyme beta-galactosidase. Lacy, encoding the enzyme permease. Laca, encoding the enzyme acetylase. Together, these three enzymes are necessary for the transport and breakdown of lactose by bacterial cells. Lactose, a sugar present in milk, is an important energy source for many bacteria. For bacteria to metabolize lactose, the sugar must be transported into cells by permease and then degraded into glucose and galactose by beta-galactosidase. The function of acetylase is not clear, although it may play a role in protecting cells from the toxic products of lactose degradation. The lac operon is regulated by a protein called the lac repressor, which is encoded by a separate gene called the Lacy gene. When bacteria are grown in the absence of lactose, the repressor protein uses helix-turn-helix motifs to bind a sequence within the lac operon promoter, P, called the operator, O. By binding to the operator, the repressor blocks RNA polymerase from binding to the promoter and blocks transcription of the Z, Y, and A genes in the operon, Figure 2.16. This is a nice way for bacteria to control their metabolism. Why expend energy transcribing genes and translating proteins if there is no lactose available for producing energy? Conversely, in the presence of lactose, the sugars act as inducer molecules that stimulate transcription of the lac operon. Lactose binds to the lac repressor, changing the shape of the repressor protein and preventing it from binding to the operator, Figure 2.16. With no repressor in its way, RNA polymerase can bind to the lac promoter and stimulate transcription of the operon. Transcribed mRNA from the operon is translated to produce the enzymes required by the cell to metabolize lactose. MICR ornus silence genes revealing a recent discovery of gene expression regulation. In 1998, scientists studying the roundworm Sinorhabditis elegans discovered small, 21 or 22 nt, double-stranded pieces of non-protein coding RNA called short interfering RNA, siRNA, so named because they were shown to bind to mRNA and subsequently block or interfere with translation of bound RNAs. For a short time it appeared that perhaps siRNAs were restricted to C. elegans, but while looking for siRNAs in other species, scientists discovered that MICR ornas, mRNAs, are another category of small RNA molecules that do not encode proteins. Genes for mRNAs have been identified in all multicellular organisms studied to date. Like siRNAs, mRNAs are regulatory molecules that regulate gene expression by silencing gene expression through blocking translation of mRNA or by causing degradation of mRNA. MICR orna genes produce RNA transcripts that are processed by enzymes, DROSHA and dicer, into short single-stranded pieces 21 to 22 nucleotides in length, Figure 2.17. The processed mRNAs then bind to a complex of proteins, called RISCs, which allows them to bind to mRNAs that are complementary to the mRNA sequence. Binding of mRNA to an mRNA sequence can either block translation by ribosomes or trigger enzymatic degradation of an mRNA. Both mechanisms inhibit or silence expression by preventing the mRNA from being translated into a protein. RNA-based mechanisms of gene silencing are generally referred to as RNA interference, RNAi. The identification of human genes that are silenced by mRNAs is currently a very active area of research. Scientists are working on exploiting RNAi as a potentially promising way to use small RNA molecules to selectively turn off or silence genes involved in human disease conditions, discussed in chapters 3 and 11. As evidence of how valuable RNAi is as a research technique, the 2006 Nobel Prize in Physiology or Medicine was awarded to Andrew Fire of Stanford University and Craig Mello of the University of the Massachusetts School of Medicine for their discovery of this method for switching genes off. 2.5 Mutations, Causes and Consequences. We conclude with a brief discussion of how genes can be affected by mutations, or changes in the nucleotide sequences of DNA. Mutations are a major cause of genetic diversity. For instance, the underlying basis of the evolution of species to develop and acquire new characteristics is governed by mutations of genes over time. Mutations can also be detrimental. Mutation of a gene can result in the production of an altered protein that functions poorly or in some cases no longer encodes a functional protein. Such mutations can cause genetic diseases. In this section, we provide an overview of different types of mutations and their consequences. Types of mutations. Sometimes mutations occur through spontaneous events such as errors during DNA replication. For instance, DNA polymerase can insert the wrong nucleotide into a newly synthesized strand of DNA, say inserting AT where AC belongs. Even though enzymes in cells work to detect and correct mistakes, errors occasionally occur during DNA replication. Mutations can also be induced by environmental causes. 4. Figure 2.17 MICR RNAs silence gene expression. Gene silencing by mRNAs is a multi-step process. 1. MICR RNA genes transcribe a primary transcript called piRNA that 2. Folds into a hairpin loop similar to the folding patterns of tRNA molecules. 3. The enzyme dicer cleaves piRNAs into single-stranded mRNAs that are 21 to 22 nucleotides long. 4. MICR RNAs bind to a cluster of proteins to form a ribonucleoprotein complex. 5. This mRNA complex can then bind to mRNA molecules by complementary base pairing with sequences that are an exact match or a close match to complementary sequences in the mRNA and mRNA. 5a. mRNA binding then silences gene expression by blocking translation or by targeting the bound mRNA for degradation by enzymes in the cell. Example: chemicals called mutagens, many of which mimic the structure of nucleotides, can mistakenly be introduced into DNA and change DNA structure. Exposure to X-rays or ultraviolet light from the sun can also mutate DNA. That glowing tan you may enjoy during the summer is not as healthy as you think. Regardless of how mutations arise, depending on the type of mutation, they may have no effect on protein production or they can dramatically change protein production and protein structure and function. Mutations can involve large changes in genetic information or single nucleotide changes in a gene, such as changing an A to AC or AG to AT. The most common mutations in a genome are single nucleotide changes, or a few nucleotide mutations, called point mutations. Such mutations are commonly referred to as single nucleotide polymorphisms, SNPs, pronounced SNPs, and they represent one major type of genetic variation in the genomes of different humans. We will discuss SNPs in several chapters. Point mutations often involve base pair substitutions, in which a base pair is replaced by a different base pair, insertions, in which a nucleotide is inserted into a gene sequence, or deletions, the removal of a base pair, Figure 2.18 on the next page. Mutations ultimately exert their effects on a cell by changing the properties of a protein, which in turn can affect traits. Figure 2.18 Types of mutations. Mutations can influence the genetic code of mRNA and resulting proteins translated from a gene. Shown here is a portion of mRNA copied from a gene, but mutations generally occur within DNA. Mutations in a gene can have different effects on the protein that is translated. Proteins are large, complicated molecules. To work correctly, most proteins must be folded into complex three-dimensional shapes. Changing one or two amino acids in a protein can alter its overall shape, dramatically disrupting its function or in some cases preventing it from functioning at all. A mutation may have no effect on a protein if it changes the codon sequence of a gene to another codon that codes for the same amino acid. Figure 2.18. This is a silent mutation because it has no effect on the structure and function of the protein. Similarly, a mutation can change a codon so that a different amino acid is coded for. Such missense mutations are also considered silent if the new amino acid coded for doesn't change the protein's structure and function. However, if the newly coded amino acid changes the structure. Career profile. Careers in genomics. It is an incredibly exciting time to consider a career in genomics. Never before have there been greater opportunities or a wider variety of career options for anyone interested in genomes. In addition to studying the human genome, scientists are actively involved in studying the genomes of many other species, including model organisms such as mice, fruit flies, zebrafish, agriculturally important crop plants and plant pests, disease-causing microbes, and marine organisms. As enormous amounts of genomic information become available, decades of work will be necessary to study what different genes do. The process of deciphering the secrets contained in genomes will involve the combined efforts of many people. Career opportunities in genomics primarily fall into four major categories: laboratory scientists, clinical doctors, genetic counselors, and bioinformatics experts. Laboratory scientists are the so-called bench scientists, because they conduct experiments at the lab bench daily. Lab scientists are often involved in carrying out experiments to discover and clone genes and study their functions. Entry-level lab scientist opportunities exist as lab technicians for those with associate's and bachelor's degrees. Higher-level scientist positions such as laboratory director positions require a master's degree or a doctor of philosophy, PhD, degree. Clinical doctors are M.D. trained physicians who conduct research and interact with patients as members of research teams. Clinical doctors may be involved in research studies to treat patients with new genetics-based treatments such as gene therapy protocols. Physicians are also important because genomics is having profound effects on medicine and will continue to do so in the future. There is an increased emphasis on translational medicine designed to bring genetics research from the lab bench to the bedside by creating new genetics-based treatments of human diseases. Genetic counselors help people understand genomics information such as how genes affect disease susceptibility. Counselor positions usually require a MS degree with training in psychology, biology, and genetics. Bioinformatics is a discipline that merges biology with computer science. Bioinformatics involves storing, analyzing, and sharing gene and protein data. The tremendous amount of DNA sequence data being generated by genome projects has made bioinformatics a rapidly emerging area that is absolutely essential to genomics. Bioinformaticians generally have solid training in both biology and computer science, and they work together with bench scientists to analyze genome data. Most positions in bioinformatics require a BS, MS, or PhD degree. Visit the Human Genome Program information page in Keeping Current, web links at the companion website for an outstanding view of career possibilities in genetics. This site also includes links to many other valuable resources. Of the protein, its function may be significantly altered. Later, we consider a dramatic example of how SNP is responsible for the human genetic condition called sickle cell disease. Sometimes mutations called nonsense mutations change a codon for an amino acid into a stop codon, which causes an abnormally shortened protein to be translated, usually creating a non-functional protein. Insertions or deletions can also dramatically affect the protein produced from a gene by creating a frameshift mutation. As shown in Figure 2.18, the insertion of a nucleotide causes the reading frame of the codons to be shifted to the right of the insertion, changing the protein encoded by the mRNA. Frameshifts often create non-functional proteins. Mutations can be inherited or acquired. It is important to realize that not all mutations have the same effect on body cells. The effects of a mutation depend not only on the type of mutation but also on the cell type in which it occurs. Gene mutations can be inherited or acquired. Inherited mutations are mutations passed to offspring through gametes, sperm or egg cells. As a result, the mutation is present in the genome of all of the offspring cells. Inherited mutations can cause birth defects or inherited genetic diseases. Acquired mutations occur in the genome of somatic cells and are not passed along to offspring. Although not inherited, acquired mutations can cause abnormalities in cell growth, leading to the formation of cancerous tumors as well as metabolic disorders and other conditions. For instance, prolonged exposure to ultraviolet light can cause acquired mutations in skin cells, leading to skin cancer. The effort to understand the genetic basis of cancer and many other human diseases is a major area of biotechnology research. Mutations are the basis of variation in genomes and a cause of human genetic diseases. Mutations form the molecular basis of many human genetic diseases. Sickle cell disease was the first genetic disease discovered. Its cause was pinpointed to a particular mutation. Figure 2.19. Sickle cell disease is. Figure 2.19 Molecular basis of sickle cell disease. A. Hemoglobin, the oxygen-binding protein of red blood cells, consists of 4 polypeptide chains. A portion of the normal hemoglobin gene is shown here along with its transcribed mRNA and the first 7 amino acids for one of the hemoglobin polypeptides, which contains a total of 146 amino acids. B. The defective gene that causes sickle cell disease contains a single base pair change in its sequence that alters translation of the hemoglobin protein. This subtle change alters the shape of red blood cells, causing them to sickle. Created by a single nucleotide change, a base pair substitution, in the gene coding for one of the polypeptides in the protein hemoglobin, the oxygen-binding protein in red blood cells. Red blood cells contain millions of hemoglobin molecules, each of which consists of 4 polypeptide chains. Figure 2.19. A point mutation in the beta-globin gene changes the genetic code so that the gene codes for a different amino acid at position 6 in one of the hemoglobin polypeptides. As a result, the amino acid valine, instead of glutamic acid, is inserted into sickle cell hemoglobin. Individuals with 2 defective copies of the hemoglobin gene suffer the effects of sickle cell disease. This subtle mutation alters the oxygen transporting ability of hemoglobin and dramatically changes the shape of red blood cells to an abnormal sickle shape. Sickle cells block blood vessels, and patients suffer from poor oxygen delivery to tissues, causing joint pain and other symptoms. Sickle cell disease is one of the best understood inherited genetic disorders. As scientists have learned more about the human genome, they have discovered that DNA sequences from people of different backgrounds around the world are very similar. Regardless of ethnicity, human genomes are approximately 99.9% identical. In other words, your DNA sequence is about 99.9% the same as DNA sequences from President Barack Obama, LeBron James, Britney Spears, Julia Roberts, Justin Bieber or virtually any other human. But because there is about a 0.1% difference in DNA between one individual and another, or around 1 base out of every 1,000, this means that there are roughly 3 million differences between different individuals. Many of these variations are created by mutation, and most of them have no obvious effects. However, Other mutations strongly influence cell function, behavior, and susceptibility to genetic diseases. These variations are the basis of differences in all inherited traits among people, from height and eye color to personality, intelligence, and lifespan. Most genetic variation between human genomes is created by SNPs. For instance, at a particular sequence, a certain base in a given region of DNA sequence from President Barack Obama may read A, Britney Spears sequence may read T, LeBron sequence may read C, and the same site in your sequence may read G. Most SNPs are harmless because they occur in intron regions of DNA. However, when they occur in exons, they can affect the structure and function of a protein, which can influence cell function and result in disease. Sickle cell disease is caused by an SNP. Refer to Figure 1.11, which shows a comparison of a gene sequence from 3 different people. An SNP in this gene in person may second have no effect on protein structure and function if it is a silent mutation. Other SNPs, person 3, cause disease if they change protein structure and function. Figure 2.20 Biotechnology is being used to detect and correct mutations. Later, we'll consider several genetic disease conditions, discuss how defective genes can be detected, and examine how scientists are working on gene therapy approaches to cure these diseases. Figure 2.20. See also Chapter 11 for more on genetic disease conditions. 2.6 Revealing the epigenome. Our study of the genomes of humans and other species has provided great insight on the epigenome and its influences on gene expression. The term epigenome refers to modifications in chromatin structure, which do not involve mutations in DNA sequence. Some epigenetic modifications are inherited, while others vary from generation to generation. Some epigenetic modifications may be reversible, while others are long-lasting. Epigenetic changes can differ between cell types in the body and in normal and diseased tissues. Diet and environmental conditions can influence the epigenome. We also know that epigenetics plays a major role in clarifying how patterns of gene expression can vary during embryonic development. The more we study the epigenome, the more we learn about how complex it is. We are only now beginning to truly appreciate the importance of the epigenome. So what kinds of chromatin modifications contribute to the epigenome? Epigenetic modifications affect both DNA and histones. For example, the addition of methyl groups, CH3, to DNA, known as methylation, commonly occurs on cytosine bases. Figure 2.21 Chromatin modifications involved in epigenetics. The chromatin modifications shown here are responsible for many epigenetic influences on gene expression. These include DNA methylation, Me, histone modification by acetylation, AC, phosphorylation, P, and methylation, remodeling of histones and other DNA-binding proteins, histone variations, and non-coding RNAs, RNAs. Methylation of histone proteins also occurs. Other examples of histone modifications include the addition of acetyl groups, COCH3, or acetylation, and the addition of phosphate groups, PO4, or phosphorylation, Figure 2.21. The extent to which methylation, acetylation, and phosphorylation affect a genome can vary greatly from one region of a chromosome to another. These chemical modifications work in complex ways to affect how DNA loops and folds in a chromosome. Such structural effects can ultimately influence how easily gene expression occurs in a particular region of a genome because epigenetic modifications affect how accessible a segment of DNA is to proteins involved in transcription. For instance, heavily acetylated regions of a genome cause the DNA to maintain a more open and less compact arrangement that allows for transcription. Removing acetyl groups from such a section of the genome typically inhibits transcription. Other epigenetic modifications that affect transcription include remodeling of histones and other DNA-binding proteins which can mask or activate regions of a genome, variations in the types of histone proteins found in DNA, and the presence or absence of non-coding RNAs that can code DNA and influence gene expression. Figure 2.21. Because the epigenome regulates gene expression, almost every major pharmaceutical company and many biotech companies are working on epigenetics projects. We know that the epigenome is involved in many diseases, including cancer. Inhibiting or enhancing epigenetic modifications to target epigenetic changes that can affect gene expression for disease treatment is a very hot area of research. For example, a relatively new category of drugs called histone methyltransferases, HMTs, which affect gene expression by adding methyl groups to histone proteins, have shown promise in treating specific malignant tumors and leukemia. Keep in mind this brief introduction to the epigenome as you learn more about genomes and ways in which the biotechnology industry is working on treatments for genetic diseases. Making a difference. Scientists around the world are working on a variety of cell therapy approaches to the treatment of human disease. For example, researchers at Rush University Medical Center and Emory University are investigating applications of a novel cell therapy using retinal pigmented epithelial cells, RPE cells, attached to tiny gelatin beads. In early-stage studies with human patients, these beads have been implanted in the brains of patients with Parkinson's disease, PD. Patients treated so far are showing improvements in Parkinsonian symptoms such as tremors, rigidity, slowness of movements, and impaired balance and coordination. This cell therapy approach holds great promise, as some of the treated patients continue to show improvements 6 years or more after treatment. The RPE cells produce levodopa, a precursor to the neurotransmitter dopamine, produced by neurons in the brain. Loss of dopamine-producing neurons is a major cause of PD. In this approach, RPE cells are cultured in a lab and attached to gelatin beads, which are necessary for the cells to survive in the brain. Then the beads are implanted into patients. The implanted cells provide a source of levodopa, which can be used by local neurons to produce dopamine. Only time will tell if this approach will become a viable treatment option or cure for PD, but without question there are many cell-based strategies underway to treat disease that will ultimately make a difference and alleviate pain and suffering.
Okay, so that concludes the complete text, chapter 2 reading. And as I scroll up, this will provide an option to pause and be able to get a better look at the diagrams. Figure 2.21, 220, figure 2.19, figure 2.18, figure 2.17, figure 2.16, figure 2.15, figure 2.14, this is actually an excellent figure. 2.13, protein synthesis. This is an excellent one. Right here, this is a very important table. Be able to tell the amino acids. Figure 2.12, eukaryotic gene mRNA processing. .11 transcription. This is also a really good one too. Pretty basic, broad, but it gives you the main ideas. Idea of semi-conservative. phosphate, sugar, base. So that's the structure of it. Phosphate, sugar, base. Those are good. Alright, and that concludes it.